as we go on with Chapter 6, I kind of group Chapter 6 and 7 together because they go together in kind of an odd way. In Chapter 6, we're going to talk about all the things prokaryotes or bacteria like. What do they need to grow? What do they like? What do they thrive in? And then how do they grow? Numerically, we're going to look at, if I start with 1 right now, how many am I going to have, in 6 or 7 hours? So that's kind of where Chapter 6 is going to take us. And then Chapter 7 is controlling microbial growth. So as we talk about what they like and how they grow, we'll talk about how do we stop them from growing. And I'm going to break a bunch of myths that most of you have when we get to Chapter 7 about what you think your cleaning supplies are doing and how you really think when you spray that Lysol on something, it's killing 99.9% of the germs. Well, what are germs? What does that really mean? So that's kind of what Chapter 7 is going to be. As we go into Chapter 6, of course, we always have a little bit of terminology we have to get out of the way when we first start. And the first term we need to understand is microbial growth. That's the name of the chapter. Okay? When we talk about microbes growing, you never should think of the cells themselves changing or getting bigger. That's not what that means. Microbial growth means these cells themselves actually multiplying. And that can happen very, very rapidly, probably more rapid than you would ever imagine it could. So I have some little animations of some bacteria growing, and these are not made-up videos. These are fancy microscopes that have video cameras on them. They injected the bacteria with some dyes so you could see them a little bit better, some fluorescent dyes, and then they just let them grow and videotaped it. They then speed up the film just so it's a little more interesting to you. So this first one is E. coli growing, and it's 440 times normal speed. Okay? So it doesn't look that impressive at first. So this is a couple hours. And keep going. Maybe 2 hours. 3 or 4 hours. Nothing magical happened. There were just some on the other side. 5 to 6 hours. 7, 8 hours. No, they don't speed up. They just grow. And so every time one divides, it becomes 2. So the more of them you have, the faster that population can grow. I like showing that video because it makes me think of, the commercials where they show you the toilet bowl under the microscope, and they show you this is after you clean your toilet, and it kills 99%, right? And there's only one little cell still sitting there. Well, what's that one cell going to look like in your toilet bowl 8 hours later? It's going to be covered with bacteria again. That's just good marketing. It's not really that important that you kill those bacteria. Really, probably in less than that, because if you use the toilet again within that 8 hours, you're putting them back in there. So that's the E. coli. And this is staph. Staph grows a little slower than E. coli, so this was sped up a little bit more. I just think this one's really neat. It kind of looks like popcorn popping. Since it's Staphylococcus, it's going to pop into a cluster. It doesn't seem quite as impressive when you watch it, but if you think about if that's growing in a boil underneath your skin, it's growing pretty fast, right? That knot's just going to swell and swell and swell. And this is, again, about instead of an 8-hour time frame, you're looking at about a 12-hour time period when that video is occurring. Okay? So what's the point? Microbial growth is impressive. Now, to get them to grow, you have to have the correct situation. Microbes are not magical. There are things that kill them, and just like us, there are things they like and things that they don't like. Okay? I personally don't like cold weather. So am I very likely to go for a 2-mile jog when it's 30 degrees outside? No. Does that mean I can't? Well, I probably couldn't, but if I could run that far, I could do it in the cold. I just don't like to. Maybe I don't move as fast in the cold. Bacteria are very temperamental, just like that. Okay. Now, another term, while I'm thinking about it, the word colony, you'll see that throughout the text. That's just when the mass, the growth of the bacteria gets so large that you can see it with the naked eye. So you know when I give you a plate or a test tube in the lab and you can see the little furry looking growth on top of it, the term for that is a colony. So don't let that bother you when you see it throughout the text. Okay. As we look at the different things the bacteria like and don't like, we're going to separate them into the physical requirements and the chemical requirements. Physical requirements are anything that you can change about the physical environment that the bacteria is in. Most important things you can alter are temperature, pH, how much salt is around. I can't think of if there's any other ones we're going to talk about in here. Those may be the only three we talk about. Chemical requirements are the things, the chemicals, the atoms, the elements that the bacteria absolutely have to grow. And these will be things like oxygen, carbon, nitrogen, oxygen. I think I said oxygen twice. Sorry. Phosphorus, sulfur, any of those things. And the interesting part of the chemical requirements we're going to look at is bacteria require almost the same exact chemical requirements that human beings do to survive, with the exception of the oxygen. We have to have that. Not all bacteria have to have that. Okay? So let's just dive right in, and we'll start with our physical requirements first. The term we're going to give to each of these different physical requirements for the bacteria, we're going to use the base word philic. Anytime you see philic in a science class, that means to like. So you've probably heard of things that were hydrophilic or hydrophobic before. You've probably heard those terms. So this hydrophilic, that means it likes water. Okay, so we're going to use that philic to help us get some terminology for things that the bacteria like. We start with temperature because this is actually one of the most useful ones that human beings use to control and look at how microbes grow. There are microbes that fit usually into three basic categories. We have psychrophiles, mesophiles, and thermophiles. Okay? The psychrophiles are the ones that like cold. And my stupid way for remembering this was always, I think it's pretty psychotic to like cold weather, just because personally I don't like it. So I could always think psycho kind of sounds like psycho, and that means cold. So I don't know. If you like cold weather, I guess it won't work for you. But that was the way I could remember that. The psychrophiles grow anywhere between 0 and 25 degrees Celsius. Now they've started using another term, a psychotroph, and it kind of fits into the middle. It's more the 15 to 25, but I don't want you to worry about that psychotroph too much. We're just going to think of the psychrophiles. They grow between 0 and 25 degrees Celsius. Now, do you guys have a true understanding of what Celsius means? It's science class, okay? So tell me what 0 degrees Celsius is. That's the freezing point of water, okay? Your freezer at home is not 0 degrees Celsius. You say, yes, it is. It makes ice. Well, it makes ice because of the way the air circulates through there. Your freezer at home is about 4 degrees Celsius, okay? Your refrigerator at home is probably about 8 to 9 degrees Celsius, okay? Would you have really thought off the top of your head that there were bacteria that preferred to grow in those temperatures? You don't like to think about that kind of thing, right? Because why do we put food in the refrigerator? Because we think it's going to keep the bacteria from growing on it, right? It does not. It's going to keep some of them from growing on it, but if you have a psychrophile that gets into your food and then you stick it in the refrigerator, that psychrophile is excited. You just put him in the place he likes to live the most, and he starts growing and growing really fast. The psychrophiles are the #1 cause of food spoilage that we have here in the United States, especially because we have that amazing technology, that refrigerator, that freezer that we can put our food in. But there are some bacteria that prefer that. Okay? So notice I gave you a range when I told you what psychrophiles like, right? On this graph down here, all the different colors, and I took the labels off on purpose. Okay? These first two little lines right here, excuse me, this second line, no, that's not right. First line and second line, I got to look at my labels. Those two triangles or curves, whatever you want to call them, that represents your psychrophiles and psychotrophs. Okay? So the psychrophile starts right here down around -8 degrees Celsius. Okay. Then the line starts going up. As that line goes up, you're increasing the rate of the growth of the bacteria. Okay? Meaning as I get warmer and warmer, the bacteria are growing faster and faster. They like it more. Okay? Then the curve peaks up here, right around 8-9 degrees Celsius. Then it starts going down until you get about to 20. Okay? So what this means is, if it's a psychrophile, it grows best at right around 8 or 9 degrees Celsius. That's its optimum temperature. But just because it prefers 8 degrees Celsius, does that mean it's going to die if you put it at 5 degrees Celsius? No. What's it going to do? It's going to grow a little slower. Still going to grow, but grow slower. So every time we talk about one of these groups, they have a minimum temperature, a maximum, and then they have a preference, the preferential temperature, the optimum, what they really like to grow in. And it kind of turns out, just lucky us, the psychrophile's optimum temperature is right around 8 degrees Celsius. That's the temperature of our refrigerator. So right now you could all go home, and I'm 100% sure we could find psychrophiles growing very, very rapidly inside of every one of your refrigerators. I don't care how clean you are. You're going to have bacteria growing in there. You just hope they don't get on or in your food. Okay? The second group, the mesophiles, they are the middle group. They like middle temperatures. The range is somewhere between 25 and 45 degrees Celsius. That means they grow from about room temperature up to body temperature and then a little bit warmer than body temperature. On the graph, it's this kind of, I don't know, yellowy orange color, the third line. They can go all the way over here to about 10 degrees Celsius. That's their minimum. But if you find their optimum, it's 37 degrees Celsius. What is that? Well, that's where they like to grow the best. But what is 37 degrees Celsius? It's the same as 98.6 degrees Fahrenheit. That's your body temperature. The mesophile's favorite temperature to grow is 37 inside of your body. Okay? But can they still grow at room temperature down here at 25? Yes, they can. These are the mesophilic group. These are the ones that cause disease. In order to cause disease, they have to be able to live inside of your body, right? So if I had a piece of food that was in my refrigerator and it had a psychrophile growing in it, is it going to hurt me if I eat that psychrophile? He's going to die as soon as he gets in my body, right? But I only care because he made my food taste bad. But if I have a mesophile that's growing on this marker right here, and then I stick this marker in my mouth without thinking about it, now I'm going to be sick because that mesophile can grow at room temperature on this marker, or that mesophile can grow inside of my body. Every pathogen we talk about in this class is going to be a mesophile. Okay? Now, one thing our body does to try to kill microbes, and we'll talk about a little later when we get to the correct chapter on immune system, is we start running a fever, trying to kill microbes living in our body. That's our body's response. But when you're running a fever, you may get up to possibly 40 degrees Celsius, but that's a high temperature. Have I killed the bacteria completely off at 40? No. But your body can slow them down enough that maybe your rest of your immune system can catch up with it. So that's the purpose of that. Okay? Then our last group are thermophiles. They like heat. They grow somewhere between 50 and 60 degrees Celsius. But again, it's kind of this pukey green color curve. They have a nice big broad range. But then there's this extra curve over here, right? That last curve, that's what we call a hyperthermophile. They like it even hotter. These two curves on the graph tell us that there are bacteria that can go all the way up to 100 degrees Celsius and just keep on thriving. Why is that 100 degrees Celsius an important number? That's the boiling point of water. To give you a reference, for those of you that like Fahrenheit, 100 degrees Celsius is 212 degrees Fahrenheit. So that means you can have a hypothermophile, And you can put him in a pot of boiling water, and you can boil him until the water completely evaporates, and you would never kill that bacteria. That's hard to imagine, right? There are bacteria that live inside of boiling lava inside of volcanoes. And we know that just because somebody decided that'd be fun to check. But there really are some living in there. Are these likely to make us sick? Probably not, because they don't really like to live down here in our body temperature. But there are some exceptions, and they have been known to be able to make us sick. But that's kind of an exception to the rule. So what would I expect you to be able to do with this? I would never ask you a specific number, okay? You need to have an idea of how the groups fit together. So if I gave you this blank graph, you should be able to tell me, okay, psychrophiles, mesophiles, thermophiles, hyperthermophiles, okay, how they fit together. The psychrophiles, like cold, they're the ones that would go in my refrigerator. The mesophiles, like the middle, they're the ones that would go room temperature or in my body. Thermophiles, like heat, they're going to be growing anything well above body temperature. Okay, so I mean, I would never give you some random number and say, okay, it's 44 degrees Celsius, what's going to be growing? That's not a good question. Okay, does that make sense? Okay, so already I'm hoping you're realizing that there is no just answer. If I ask you what temperature do bacteria like, the answer is it depends on the bacteria. There's a really broad range of what they prefer to grow in. Okay. The next topic is pH. Okay, and I need to know kind of what you guys understand about pH. So what is neutral pH? 7's neutral. So if I go down below 7, so the range goes from, not 0, sorry, from about 1 to 14. 7's in the middle, that's neutral. So if I go down from 7 towards 1, what do we call that? That's becoming more acidic. Good. Low pH is acidic. If we go up above 7 towards 14, what do we call that? Basic or alkaline. Some of you may have heard that before. It's becoming more basic, higher pH. Okay? Most things inside the human body, with the exception of the stomach, are right around neutral pH. Most things are right around 7. It turns out that bacteria kind of have that same preference. Most bacteria like the 6.5 to 7.5 range. So if you dump most bacteria into something really acidic down to a pH of 2, it'll kill most bacteria. Is that good for us? Because one of the most common entryways for bacteria into our body is through our gastrointestinal system. And if it makes it down to our stomach, it's going to die because the pH of our stomach is about 2. Okay? Same thing goes if you make it high pH, bacteria really don't like that. But there's always an exception to the rule. There are a few bacteria that love to grow down here in the acidic range on the pH scale. They love low pH. Okay? The word we give to those bacteria, that group, is acidophile, the acidophiles. They like acid. Okay? So I have a little question up here. See if you guys can think for me. Can you think of any food you have at your house that an acidophile may be growing in it right now? So before you answer, let me give you a little bit more information. Okay, if you know, just be quiet for a second and play along. Okay, you got to think about, are there any foods, anything in your kitchen that you put into something acidic to help make the food and preserve the food? Any ideas? I couldn't hear you say it loud. Salt's not acidic. Lemon juice, pickles, any of those fruit juices, orange juice, lemon juice. If you have that at home, if it's in anything clear, pick it up and swirl it and look at the bottom and see what happens. Pickle jar. If you have a jar of pickles at home, go home, pick it up out of the refrigerator, hold it above your head in the light where you can see it, and swirl it. You know how in lab when I swirl the test tubes and I say, do you see that? It looks like snot in the bottom of the test tube? Guarantee you, if you go home and swirl a pickle jar, you will see the little twist, the little white stuff in the bottom. That is acidophiles. How many of y'all drink pickle juice? Yeah, like half the room is like, heck yeah, right? So, since half of you drink pickle juice, are the acidophiles going to hurt you? Obviously not, or half of y'all would be really sickly right now, right? They just grow in the acid. But as soon as they get into your body, is it that acidic anymore once it leaves your stomach? It's not that acidic anymore. Your pH is going to kill them. I just always think that's kind of neat to think about. And if you want to gross somebody out, take their pickle jar and show them. Swirl, and don't tell them it's in everybody's. Just swirl and tell them that they have snot, bacteria growing in their pickle jar. Okay? It really only becomes a spoilage issue with your fruit juices because then they start breaking down the sugars that are in the fruit juice, and that gives you a more of an alcoholic taste to your fruit juice, and it gives it a spoiled taste. Your pickles are really never going to spoil with the acidophile, but your fruit juice spoils because of the acidophile. Okay, woke some of y'all up now. Got y'all listening to me. Good. All right, well, I didn't wake you up. That's all right. All right. Osmotic pressure. Now, I should be able to say, I know you guys all know what this means because we just had a test on it, but let me just assume that some of you may not know what this means. Osmotic pressure refers to the amount of solute or salt concentration you have. Okay, so on our last test, we learned that if you took a bacteria that was 1% salt on the inside, right? I think this was your actual test question. And you put it in a solution that was 5% salt on the outside, what did you just make, a hypotonic or hypertonic solution? Uh-oh. Hypertonic solution. See, there are reasons I make you guys learn these things because you're going to keep seeing them creep up and bother you, okay? This is now a hypertonic solution. There's more salt on the outside. If there's more salt on the outside, where's there more water? Inside of the cell. So the water is going to do what? Move out of my cell. So we learned that. Well, we relearned that in Chapter 4, right? Well, here it comes sneaking up again. Most bacteria only like an isotonic environment. What does that isotonic mean? No net movement, a balance, right? Where you have the same amount of salt on the inside of the cell that you have on the outside of the cell. All bacteria have some salt in them. So they like to be in some salt. They don't even really like to be in pure water. They like a little salt. But most bacteria, if you dump extra salt on the outside of them and you make a hypertonic environment, that bacteria is going to leak all of its water out. It's going to shrivel up and it's going to die. And I think we even talked about this a little. We use that a lot, right? We dump all kind of salt in something, and we know that that's going to keep something fairly fresh. That's what we do in almost all of our canned foods. We just dump salt in it. That way we don't have to worry if it's got bacteria in it or not. If there's bacteria in it, they'll just leak their water out and they'll die. And then you'll just eat dead bacteria parts. Won't hurt you. Okay? But there's always that bacteria that just keeps living no matter what we do to it. There are bacteria that you can dump not as much, but a lot of salt on the outside of him, and he just keeps on growing. This group of bacteria is called halophile, halophilic. Halo comes from where your salt elements are on the periodic table. Is why it's a halophile. Don't let that bother you, okay? Excuse me. There are two types of halophiles. There are facultative halophiles and obligate halophiles. You're going to see these two words again. So you can figure out what this means. If I am obligated to do something, what does that mean? I absolutely have to do it. So if it's an obligate halophile, that means he absolutely has to have a high salt environment to live in. Okay? These aren't bad for us. They only live in salt. If you take them out of salt, they die. Okay? So there's kind of some weird ones. The ones that are dangerous to us as far as especially food preservation, are the facultative halophiles. They can go either way. If they're hanging out and living happy, free, no salt around them, they don't care, they're good. You could dump a whole can of salt on them, and they just smile and keep on growing. They don't care either way. So if you get a facultative halophile contaminant in one of our big factories that we produce canned foods in, and you get that bacteria, that facultative halophile into those cans, when you dump all the salt in, it doesn't kill them. And then everybody comes along and eats that can of food thinking that the salt has killed the bacteria, and that bacteria just keeps on growing. Do any of you eat sushi? I do. I'm not telling you not to. But one of the things that, this is more American than other cultures, but one thing that most of your Americans do to the sushi, to fish, is we salt our fish. Why do we salt it? To kill the bacteria. Mainly because we can't get it quite as fresh as some, we can't here in Mississippi, but some places can't get the fish quite as fresh, and certain types of fish, you have to import. So a lot of it we salt to try to kill any bacteria that's growing on it. But what happens if a facultative halophile is growing on it? It doesn't care if you put salt on it. You can get some pretty gross food poisonings from eating something that a facultative halophile is spoiling. Excuse me, my voice is hanging in there. I'm going to make it. Okay? So those are our 3 physical requirements. They all have a certain temperature they like, all have a certain pH they like, and they all have a certain salt concentration they prefer to grow in. Okay? So now let's look at our chemical requirements. And I have a little acronym that hopefully will help you remember your chemical requirements. And the acronym is CHONPS, CHOPS. Yeah, I know it's not a real word, but it was the best I could come up with to help you have some way of remembering, having a key that would help you remember these required chemicals that all life on Earth has to have. Since bacteria are alive and they live on Earth, they have to have these chemicals to survive. The C and the H stand for carbon and hydrogen. So we're not going to have chemistry class, but you should all be able to tell me the reason why do they have to have carbon to survive? Why does a bacteria need carbon to survive? I don't really have anything to do with electrons. That sounded like a smart answer there. Oh, it is a waste product. We learned that in Chapter 5, but it's a waste product because we took it in. Why do we have to take that carbon in? Because we are from Earth. Have you ever watched the Sci-Fi Channel? Y'all need to expand your brains, expand your horizons, watch Sci-Fi Channel. All human life, shouldn't say human, all life on Earth is carbon-based. Y'all heard that before, right? What does that mean? That means every molecule in your body has a carbon backbone. Does DNA have carbon in it? RNA? Proteins? Fats? Sugars? Every one of those molecules, you know you have to have those to live, right? Every one of those molecules has a carbon backbone. We draw them. Every one of them, you're going to see a C connected to a C connected to a C. They all have to have that. So if you're going to be able to, if I'm a bacteria and I'm going to be able to reproduce myself and make a new me, I'm going to have to make more DNA and RNA and proteins, right? So I'm obviously going to have some way of getting carbon so I can make those molecules. The hydrogen is just kind of a fact of science. Everywhere you got carbon, hydrogen's just coming along for the ride. And that's where you guys gave me the answer, electron. Because the two of them hanging out together, that's to satisfy your electron balance, your charges. So you probably learned that in some chemistry class somewhere, and that's stuck in your brain. So you didn't really give me a wrong answer. You gave it to me too soon. Okay? So that's just one of those facts of life. If it's alive on Earth, it's got to have carbon. Let's skip the O. We'll come back to it. Now let's go to the N. N stands for nitrogen. Now it was a little harder, but the answer is easy. It's not going to pop in your head quite as easy. But guess what molecules you have to have nitrogen to make? DNA, RNA, proteins, fats, sugars. Seen a trend here? Every one of those molecules that we have to survive, not only do they have to have carbon and hydrogen in them, they also have to have nitrogen. So if I'm a bacteria and I want to make more of myself, I got to make more DNA, got to make more RNA. I've got to have more of that nitrogen so I can make those molecules. The P, phosphorus. Same thing. You need it to make DNA, RNA, proteins, sugars, fats, all of those. But you need that P for something else too. What else do you need P's to make? ATP, which is energy. If I'm a living thing, and bacteria are alive, they have to have ATP. So they have to have phosphorus to make that. The last little guy on the end, the S, sulfur. You don't have to have quite as much of that. It's only required in some of your proteins. Use it to make 2 amino acids, cysteine and methionine. Kind of some weird guys, but you got to have that if you want to make a protein. So I used to have a big long slide that said what you need each of these for, but I don't think you need that because what's the answer? You need all of these molecules to make all the macromolecules you need to survive, the DNA, the RNA, and I don't think that's something you should have to memorize, right? Because you all know you have to be able to make more DNA to live, right? Bacteria do too. So it's not something you even really need to study. The only one of these chemical requirements I think we need to talk about a little bit is the O. And the O stands for oxygen. But oxygen is one of these that we kind of got to put some question marks by. It is the only one of the chemical requirements that is not required by all microorganisms to survive. Now, human beings, yes, oxygen is not a question mark. We have to have it. But if I'm a bacteria, I may not need that oxygen. We've already talked about this a little bit, right? How do we talk about that? Fermentation, right? We said in Chapter 5 there's some bacteria that if you take that oxygen away, they don't care, right? They just stop doing electron transport. They stop doing Krebs cycle. They do glycolysis, and they ferment to make energy to survive, right? So you already know there's some that can live with it and some that can live without it. So let's look at the proper terminology. On Earth, there are microbes that are called obligate aerobes and obligate anaerobes. So tell me what an obligate aerobe is. Has to have oxygen. Obligate aerobes are like humans. If you take oxygen away from those bacteria, they will die just as quickly as a human being would if you deprived it of oxygen. So what would an obligate anaerobe be? Absolutely has to live in the absence of oxygen. So if you put an obligate aerobe right here on the desk, what would it do? It would die because there's oxygen in the room, right? Are those really harmful to us? No, because are we ever going to be somewhere that guy's living? Yeah, no, because we wouldn't be living there, right? So we don't have to worry about the obligate anaerobes. The ones we have to worry about are these facultative anaerobes because they can live with it or without it. So if they're hanging out in this room right here, heck yeah, oxygen sounds good. I will breathe in some oxygen, I will make me some ATP, and I will live. If you take it out of this room and you put it inside of a jar and you seal it off and take his oxygen away, what's he going to do? Start fermenting. He don't care. He just switches to his other metabolic pathways and he keeps on living. So if oxygen's there, he's going to use it. If it's not there, he's going to figure something else out. He's going to ferment. Excuse me. There's two other little groups here, though, and these are kind of weird guys. There's something called an aerotolerant anaerobe. So let me help you figure out how to remember this. If I tolerate somebody, what does that mean? Right. So if I tell you, yeah, I tolerate Kelly, that means I don't really like her. But if she's around me, I'm going to just deal with it. So an air-tolerant anaerobe, does he like oxygen? So is he going to use it? He's not going to use it. But if you put him in this room where oxygen is, can he live? Yes. So you have to kind of think of it that way. It's a bacteria that can tolerate oxygen, but he can't use it. What is different about an aerotolerant anaerobe and a facultative anaerobe? Facultative ones will use it if it's there. Aerotolerant just tolerates it if it's there. Does that make sense? Understand the difference? The microaerophiles are kind of weird guys. They need oxygen, but they don't like quite as much as what is inside of normal air. So we don't really deal with those a whole lot in this classroom. I'm not going to test you on those, but it's in all your figures. So I didn't want somebody to ask me, hey, what's that other guy on there? And there's always somebody that would. So I don't really want you to worry about him too much. Now, from what you know about me so far in this short period of time, you probably have already figured out I like pictures and graphs, right? I'm a very graphical person. So I'm very likely to not just ask you to give me a definition, but I'm going to ask you to tell me how would these three grow in the same situation. And that's what this picture is. So I gave you a test tube. It's got media in it, some liquid media that bacteria grows in. You put the bacteria in it and you seal the top of it. Right away, in one of these test tubes, where would the most of the oxygen be? In the air that's in the top, right? Eventually, I know that would go away, right? But we're talking about right at the beginning. Okay? So which group's going to be hanging out right here at the very top of the liquid? Why are the obligate aerobes all at the top of the broth? They have to have the oxygen so they're close to the top, right? Over here to C, why are all these guys at the bottom? Oxygen will kill them. They want to be as far away from oxygen as possible, so they're going to hide down on the bottom. Okay? Now, what's the difference between B and D? Looking just at the pictures, what's the difference? B has a cluster at the top, and then some throughout, right? D is just evenly dispersed. In the B picture, the clump at the top, what are they doing? They're using the oxygen. What are these other ones doing? They dead? They're not going to fight the crowd at the top, right? Because a facultative anaerobe doesn't have to have the oxygen. So if there's a big crowd up there trying to use the oxygen, they'll just kind of scoot down and hang out and ferment down here in the bottom somewhere where they don't need the oxygen. Aerotolerant in the D part of the picture, they don't use the oxygen, so they just spread out and get lots of room so they can grow however they want to grow. Does that make sense? I like pictures. All right. So where we're going to be going with this now is looking at a number of how microbes grow, giving it a numerical value. Okay? And it's not hard math. It's like third grade math. Okay, I promise. It's really not hard math. You can all count to 2, right? No, you did not all shake your head. It shows you're not listening to me. You can all count to 2. So I know this is math you can all do. So don't say, oh my God, I hate college algebra. I do too. We're not doing college algebra. We're just going to do easy math to talk about how bacteria grow. Okay? We're going to talk about bacteria as a culture. What does that word mean? It's just a group of bacteria, right? We can't really just look at what one is doing without thinking about how the overall environment and the group of them kind of affects this. But before we get there, let's look at what goes on with each one bacteria as growth is occurring. Bacteria and all prokaryotic cells grow by binary fission. The process of binary fission is not complicated. You all learned mitosis at some point, right? Prophase, metaphase, anaphase, telophase, all that mess. You ain't got none of that in binary fission. Binary fission is about 4, and I guess it could be stretched to maybe 5 simple little steps. In binary fission, You always are going to look at what's going on to this one cell. The first thing that has to happen in binary fission is your cell elongates. It stretches out. That kind of makes sense, right? What happens to women when they're making a new life? They stretch out, right? Making room to grow the new life in there. So the cell has to stretch out a little bit. After the cell stretches out, the bacterial cell replicates its DNA. Why? You made a new one, right? Because the original one and the new one have to both have the exact same DNA, right? So he just makes an exact copy of all of his DNA. After he makes his exact copy, you move down to what is step 2 in the picture. The cell wall and the plasma membrane start making an invagination, meaning they kind of start to pinch off. And it sequesters one piece of DNA into one side and the other piece of DNA into the other side. Just keeps on going until the cell wall eventually forms a complete, what they call cross wall, just a separation of the two pieces of DNA. Once that wall forms completely, the two cells will separate, and these two cells are identical to the original cell. Isn't that a whole lot easier? Okay. So let me throw a little bit of terminology at you that we're going to need to understand the next class. It's not letting me write on the screen. That's okay. All right, that very first cell, the original one, that is called the mother cell. Okay? It's also considered generation 0. And that tricks a lot of people up. They want to call it, since it's one cell, it's generation 1. That's not the way we use the word generation, though, is it? If your parents had you, you're the first generation. Then when you have kids, your kids become the second generation. Okay, so we're just using terminology you use normally, just sticking it on some bacteria. Okay, so the original cell, that's the mother cell, that's generation 0. When that cell divides and makes two new ones, both of these cells are now generation 1. Generation 1, all right? And they are called not mother cells, but daughter cells, okay? So one mother cell at generation 0 makes two daughter cells at generation 1, first generation. If we went another generation, how many cells would we have? 4. Then we would be at generation 2. So let's see if you can kind of figure out where this is going to go. And I'm going to write it so I can keep up with it. I'm not good at math. Okay? So we started out with one mother cell, and that was generation 0. That one cell became 2 cells. That was generation 1. 2 cells became 4 cells, generation 2. 4 cells will become how many? 8, 16. Y'all gave me lots of answers. What's the answer? Think about it. Draw it. 1, 2, 3. I got 4, right? He'll become 2 more, 2 more, 2 more, 2 more. The answer is 8. But I wanted you guys to see where the answer comes from. It's not about memorizing something, okay? So I got 1, 2, 3, 4, 5, 6, 7, 8 cells, right? I'm doubling every time. So from 4 I go to 8, and that's generation. So 8 cells will turn into 16, generation 4. 16 becomes 32 is generation. 32 becomes 64, generation. 64 becomes 128, and that's generation. What comes next? Come on, I know at least one more. 256, generation. All right, so what point am I making now? It doubles every time. And once we've gone through about 8 generations, what do we need? A calculator to figure it out, right? So microbiologists, we're not mathematicians. So if there's an easy way to write something mathematically where we don't have to get these big old numbers, we take advantage of that. This type of growth, where you double your number of cells from each generation, is called exponential growth. Okay? Do y'all know what an exponent is? So a little number that goes at the top of another number. That's superscript, right? Okay. Let me show you a really neat trick. The generation number can always be used as an exponent. And you can always use the base number 2 to figure out how many bacteria you have. Okay, so let me prove it to you. I usually mess this up because I'm not good at math. So why am I using a base number 2? Because one cell always becomes 2 cells. With a base number 2, 2 to the generation, so 2 to the 0 is 1. 2 to the first power is 2 to the second power is 2 to the third power, 8. Okay? If you don't believe me, go home and get you a calculator and do 2 to the third power and see what happens. If you keep going, 2 to the fourth power, that's 16. 2 to the fifth power, 32. 2 to the sixth power, 2 to the seventh power, 2 to the eighth power, and so on and so forth. So we don't have to use these big horrible numbers as long as we can always remember what these other numbers tell us. Okay? So let's make sure you remember. The base number 2 tells us that one cell always becomes 2 cells. What is the exponent always going to be? Generation number. And then if you work that out, you get the answer of how many cells you have. Does that make sense? Okay. Where we're going to go with this is figuring out how many cells we have at a certain amount of time. Because every time you go from 1 cell to 2 cells, it takes a certain amount of time. And that's called the generation time. For some cells, a generation time is 10 minutes. So in 1 hour, you've gone through 6 generations. You would have 2 to the 6 cells. See how much easier that is than trying to figure out numbers? And we're going to practice with it. But, okay, generation time, how long it takes for 1 cell to become 2 cells. Sometimes generation time is 1 hour. Okay? So in a 24-hour period, I got a whole lot of cells, right? How many cells? Just to show you the numbers. I put this chart in your notes just so I could show you because I can't go much above 8th generation math in my head. Look at what 2 to the 20th power is. 1,048,576 cells. Okay? So if you had 1 bacteria left in your toilet bowl after you cleaned it with some Lysol, right? And let's say that bacteria is pretty slow. His generation time's an hour. By the end of the day, that 1 bacteria is now more than 1,048,576 cells. That's kind of hard to believe when you just hear the numbers, isn't it? But they grow that fast. Okay? So I want to stop here. Let me show you where we're going with this. We're going to work some word problems. And it's not going to be bad, I promise, because I don't like math, and I'll say it 100 times. I don't care if I'm recording myself. We're going to do some word problems, and then we're going to look at some graphs. Okay? And this is not going to be bad, but I want you to make sure at least go through and kind of read through this exponential growth part in your textbook. Make sure this makes sense. If when I write a 2 to the 5th power, you have no clue what that means, just do a little bit of reading. The math's not going to be bad, but you may have to do a little bit of work on your own. Does anybody have any questions about what we did today?